The rumen is composed of 4 different compartments: the rumen, reticulum, omasum, and the abomasum. Feed travels through the esophagus into the rumen, which is the first of the 4 compartments. The function of the rumen is the digestion of fiber through anaerobic bacterial fermentation. These anaerobic microorganisms live and reproduce within the rumen. Volatile fatty acids such as acetic, propionic, and butyric acids are microbial byproducts which are absorbed through the many papillae that line the internal surface of the rumen. The digesta separates into 3 different layers. Partially digested hay settles to the bottom, followed by the liquid mixture of water and saliva. Methane and carbon dioxide gases released from anaerobic respiration rise to the top of the rumen, allowing the liquid mixture to flow through the reticular rumen.
The smaller of the 2 sections of the reticular rumen and the second compartment in the digestive cavity is the reticulum. Anatomically, the interior of the reticulum is covered with a highly structured epithelium folded into flaps that form polygonal cells, giving it a reticular honeycombed appearance. As digestion proceeds in the reticular rumen, feedstuffs are reduced to smaller and smaller sizes while microbes continually proliferate. In many respects, the reticulum is considered part of the rumen because food particles are free to flow between the 2 organs. The smaller and more dense materials tend to be pushed into the reticulum for maximum absorption of volatile fatty acids through fermentation. Ruminants are constantly either eating or cud chewing. For this reason, most of the animals are not very selective in their choice of food. Consequently, many animals mistakenly ingest potentially harmful metal particles. As a form of prevention, ruminant managers can have a magnet lodged into the reticular lining to collect metal scraps for later removal. The rest of the foodstuff is then passed to the omasum through a short tunnel for further digestion.
The omasum is the 3rd compartment and is lined with folds of tissue arranged in layers or pages in a book. Each fold has a papillae on its surface that is used to absorb the excess volatile fatty acids that were not absorbed by the rumen. When the digested remnants reach the omasum, they are in a liquid form, making it easy for transport. This movement happens very quickly, and the omasum contracts to push the remnants through to the abomasum. While this happens, the papillae also absorbs water and electrolytes to dry out the remnants before passing to the 4th compartment. The water and electrolytes are absorbed through the bloodstream for further use to the body.
The abomasum of suckling calves is a source of rennet, an enzyme commercially used in cheese production. For this reason, it is also called the rennet bag. The abomasum is the 4th and final stomach compartment. It is the most similar in function and location to the stomach of non-ruminant animals. Also referred to as the true stomach, it is the only compartment with glands that secrete acids and enzymes used to break down nutrients, especially the acid hydrolysis of proteins. The digesta then travels into the small intestine for further digestion and absorption before leaving the body. The normal location of the abomasum is along the ventral midline, slightly off to the right side of the animal, between the 7th and 11th ribs. In some cases, the stomach fills with gas, causing the abomasum to be displaced upwards on the left side of the body, potentially leading to further complications such as bloat. And now a message from Dr. DePeters.
A high forage diet tends to produce more methane. A high concentrate diet tends to produce less methane. So those types of diet strategies in the future will be used to modify the emissions of greenhouse gases that come from ruminants. Okay, the next thing we're going to do is we're going to take a look inside of the rumen of Edwina. So we always put gloves on, so we use some latex gloves. And if you could smell through the camera, what you would smell is you would smell all of the volatile fatty acids that are being produced. So the acetic, the propionic, and the butyric acid that are being produced by the anaerobic fermentation have a very pungent smell. I mean, they're volatile fatty acids, meaning they're volatile, and you can smell them. And if we look, we can see alfalfa hay, the fiber component, and that's what makes Edwina's rumen unique. You can see all the rumen fluid that contains the millions and millions of anaerobic bacteria. And in the current environment, when we're talking about greenhouse gases, we can study methane, we can study hydrogen sulfide, so we can look at the differences if we were to add some kind of rumen modifier. We talk about many different compounds that may change the microbial population. How does that rumen modifier change the microbial population, change fiber digestion, and change the amount of methane that's produced by Edwina.